So in this video we are going to learn about respiratory system, trachea, bronchioles and bronchi. So we could see a picture where trachea is shown, main bronchus region, along with that segmental bronchus, then bronchioles, then terminal bronchioles, along with that respiratory bronchioles, conducting portion, and along with that respiratory portion is shown separately. The conducting portion is made up of nasal cavities, nasopharynx, larynx, trachea, bronchi, and bronchioles. The trachea branches to give rise to two primary bronchi. This then branch successively to give rise in turn to secondary and tertiary bronchi. These then branch to give rise to several orders of progressively smaller airways called bronchioles, the smallest of which are called terminal bronchioles. These are the last components of the conducting portion of the respiratory system. Then terminal bronchioles give rise to respiratory bronchioles, which ultimately lead to the alveoli. Then trachea. The trachea is a wide, flexible tube, the lumen of which is kept open by 20 tracheal cartilage, which are C-shaped rings of hyaline cartilage. The gaps between the rings of cartilage are filled by the trachealis muscle, a bundle of smooth muscle and fibroelastic tissue. Together, this hold the lumen of the trachea open but allow flexibility during inspiration and expiration. The respiratory mucosa and submucosa are adapted to warm and moisten the air and to trap particles in mucus. Then mucosa and submucosa of trachea. The respiratory mucosa is made up of epithelium and supporting lamina propria. The epithelium is tall columnar pseudo stratified with cilia and goblet cells. The supporting lamina propria underneath the epithelium contains elastin that plays a role in the elastic recoil of the trachea. So when we learn about the trachea is a wide flexible tube, the lumen of which is kept open by 20 tracheal cartilages, which are C-shaped rings of hyaline cartilage. The gap between the rings of cartilage are filled by the trachealis muscle, a bundle of smooth muscle and fibroelastic tissue. Together, this hold the lumen of the trachea open but allow flexibility during inspiration and expiration. The respiratory mucosa and submucosa are adapted to warm and moisten the air and to trap particles in mucus. Then mucosa and submucosa of trachea. The respiratory mucus is made up of epithelium and supporting lamina propria. The epithelium is tall columnar pseudostratified with cilia and goblet cells. The supporting lamina propria underneath the epithelium contains elastin that plays a role in the elastic recoil of the trachea during inspiration and expiration, together with blood vessels that warm the air. The submucosa contains glands that are mixed seromucous glands. The watery secretions from the serous glands humidify the inspired air. The mucus, together with mucus of the goblet cells, traps particles from the air which are transported upward against the pharynx by the cilia on the epithelium. This helps to keep the lungs free of particles and bacteria. This is a cross section through the trachea showing the major layers. Here we could see a cartilage portion. Along with that, submucosal region, then smooth muscle is there. Then next picture. This is a higher power image of trachea showing the glands and epithelium in most detail. Blood vessels are there. Then lumen, lamina propria, along with that glands, smooth muscle, etc. Then bronchi. We could see glands, cartilage, etc. are associated with that lumen region. Along with that, smooth muscle is there. The trachea branches into two primary bronchi, which branch into secondary and then tertiary bronchi. In the tertiary bronchi, there is less cartilage and it does not completely encircle the lumen as shown diagrammatically above. Notice also how the mucosa is folded and think about how this might change as you breathe in and out. This is a picture of tertiary bronchus. Compare this picture with that of trachea. Then when we try to look at this picture, we could see cartilage region, lumen of tertiary bronchus, then smooth muscle, etc. Then bronchioles. The tertiary bronchi branch into bronchioles, which have a diameter of 1 mm or less, and the wall structure changes. The epithelium is made up of ciliated columnar cells in larger bronchioles or non-ciliated in smaller bronchioles. Difficult to see at this magnification. Then there are no goblet cells, but there are cells called Clara cells. The cells are secreted, they secrete one of the components of surfactant. Then asthma. Because the diameter of bronchioles is reliant on smooth muscle tone, these airways can almost completely shut if smooth muscles contract strongly, which can happen in an asthmatic attack. Then terminal bronchioles are there. Before that, this is a picture of a bronchiole. So there is no cartilage and no glands. Can you identify the ring of smooth muscle? Okay, so these are smooth muscles associated with that part. Then bronchiole is there. Then terminal bronchioles. The final branches of the bronchioles are called terminal bronchioles. These have a layer of smooth muscle surrounding their lumens. Then stimulation of the vagus nerve, parasympathetic, cause the smooth muscle to contract and reduce the diameter of the terminal bronchioles. Then small sacs are found extending from the walls of the terminal bronchi called respiratory bronchioles that are lined by a ciliated cuboidal epithelium and some non-ciliated cells called Clara cells. Then this picture of a red color in the right side shows terminal bronchiole. Note that this is at a lower magnification than the three pictures. This is the last part of conducting portion of the respiratory system and has smallest dimension of or less than 1 mm. There is no cartilage, no glands, and some smooth muscles is still present, and there is no goblet cells. The epithelium is either columnar or cuboidal. So when we learn about lungs, let's have an introduction about this topic. The lungs are a pair of primary organs of respiration present in the thoracic cavity beside the mediastinum. They are covered by a thin double-layered serous membrane called the pleura. Then the respiratory system consists of two components, the conducting portion and the respiratory portion. The conducting portion brings the air from outside to the site of respiration. Then the respiratory portion helps in the exchange of gases and oxygenation of the blood. The conducting portion of the respiratory system includes nose, nasopharynx, pharynx, larynx, trachea, and a whole series of successive narrowing segments of bronchi and bronchioles. The conducting portion ends at the terminal bronchiole. The respiratory portion begins from the respiratory bronchiole and continues with the alveolar ducts, alveolar sacs, and finally ends at the alveoli, where the significant exchange of gases take place. The branching pattern of these conducting passages looks like the branching of a tree and hence called the tracheobranchial tree. The conducting portion of the respiratory system include the nose, nasopharynx, larynx, trachea, and a whole series of successive narrowing segments of bronchi and bronchioles. The conducting portion ends at the terminal bronchiole. The respiratory portion begins from the respiratory bronchiole and continues with the alveolar ducts, alveolar sacs, and finally ends at the alveoli, where the significant exchange of gases take place. The branching pattern of these conducting passages looks like the branching of a tree and hence called the tracheobranchial tree. Then the right lung has 3 and the left lung has 2 lobes. Each is aerated by a secondary or lobar bronchus. The lobes are further divided into smaller pyramidal shaped sections called the branchiopulmonary segments. There are 10 branchiopulmonary segments in each lung with their apex directed towards the hilum. There are 10 bronchopulmonary segments in each lung with their apex directed towards the hilum, and each is aerated by a tertiary or segmental bronchus. The alveoli are the structural and functional units of respiratory system. There are around 300 million alveoli in an adult human, amounting to approximates 80 square meters of surface area for the gaseous exchange. The lungs are an essential component of the pulmonary circulation, where the deoxygenated blood pumped by the right ventricle of the heart is gushed through the pulmonary arteries to alveolar capillary beds of the lung for gaseous exchange. The oxygenated blood from the capillaries of the lungs is returned to the left atrium by the four pulmonary veins. Then, issues of concern. These are not actually the issues of concern, but some of the unique features of the lung. The early prenatal development of the lungs depicts the development of an exocrine tubule alveolar gland. Initially, it begins an offshoot from the endoderm of the foregut as a lung bud. Later, branches to form the tracheobranchial tree and canalizes. This is precisely how the glands develop. Hence, initial development of the lung is said to be pseudoglandular development. The lung alveoli start developing in the prenatal stage and very important in a live birth with the active secretion of surfactant from its type 2 pneumocytes. Nevertheless, 95% of alveoli are formed postnatally during the first 8 years of life, that too a majority being in the first 3 years. The lymphoid tissues of the tracheobranchial system is modified to contain diffuse, aggregated, and solitary lymphatic nodules specialized to be called bronchus-associated lymphoid tissue or BALT. This is important for hypersensitive reaction which occurs with respiration. They also have significant role in the defensive mechanism from the exposed microorganisms from reaching the lungs through the inhaled air. The conduction portion of lung begins at the trachea and extends to the terminal bronchioles. Outside the lungs, the conduction system consists of nasal cavities, nasopharynx, larynx, and trachea. Within the lungs, the conducting portion is splits into paired main bronchi. The bronchi begin as branching pattern, splitting next into lobar or secondary branchial branches and then again into segmental bronchi. The tertiary bronchi continue to divide into small bronchioles, where the first change in histology take place as cartilage is no longer present in the bronchioles. The end of the conduction portion of the lungs is at the first segment called the terminal bronchioles. The terminal bronchioles open into the respiratory bronchioles. This is the start of the respiratory portion of the lung. The conducting portion provides the pathway for the movement and conduction of the air entering the lung. Specialized cells collaborate to warm, moisturize, and remove particles that enter. The cells are the respiratory epithelium and comprise the entire respiratory tree. Most of the respiratory epithelium is ciliated pseudostratified columnar epithelium. The following 5 types of cells are in this region, like ciliated cells, goblet cells, basal cells, brush cells, neuroendocrine cells. The ciliated cells are the most abundant. They control the actions of the mucociliary escalator, a primary defense mechanism of the lungs that remove debris. While the mucus provided by the goblet cells traps inhaled particles, the cilia beat to move the material toward the pharynx to swallow or cough out. Goblet cells, so named for their goblet-shaped appearance, are filled with much granules at their apical surfaces, with the nucleus remaining towards the basilar layer. Goblet cells decrease in number as the respiratory tree gets progressively smaller and are eventually replaced by club cells, previously Clara cells, when they reach the respiratory bronchioles. The basal cell connect to the basement membrane and provide the attachment layer of the ciliated cells and goblet cells. They may be thought of as the stem cells of the respiratory epithelium, as they maintain the ability to potentiate ciliated cells and goblet cells. Then brush cells, occasionally referred to as type 3 pneumocyte cells, are sparsely distributed in all areas of respiratory mucosa. Brush cells may be columnar or flask-like and are identified by their short microvilli covered apical layer resembling a push broom or appropriately a brush. No function has been officially assigned to the brush cells, though there are many proposed mechanisms. One popular proposal suggests they have a chemoreceptor function monitoring air quality due to their association with unmyelinated nerve endings. The bronchial mucosa also contains a small cluster of neuroendocrine cells, also known as Kulczycki cells. I'm not sure about the pronunciation. They have neurosecretory type granules and can secrete several factors. This include catecholamine and polypeptide hormones such as serotonin, calcitonin, and gastrin-releasing factors like bombicin. Like brush cells, these neuroendocrine cells make up only a small portion of mucosal epithelium, around 3%, within the bronchial submucosa or submucosal glands. These glands are composed of a mixture of serous and mucinous cells similar to salivary gland tissue. The secretions are emptied into ducts and then on the bronchial mucosa. Although individuals may show oncocytic metaplasia of these glands. Smooth muscle bundles are present at all levels of the airway to allow for regulation of airflow. There are progressively fewer smooth muscle fibers processing from bronchi to alveoli. When we talk about functions, respiratory functions of lungs, primary function is respiration. We know that. The lungs are the primary organs of respiration where the exchange of gases take place. In the alveoli, there are the functional units of lungs. The oxygen is taken up, the carbon dioxide is removed from the bloodstream through the alveolar capillary bed. Along with that, air conditioning is happening. The function of conducting part is not only to lead the air but also act as an efficient air conditioner. This conditioning is done by warming or cooling the inhaled air to bring it to the level of body temperature, humidifying the air, and also the removal of all the foreign particles present in it. The removal of foreign particles like dust, bacteria, virus, etc. is done by mucus secretion, which traps the suspended particles and bearing of the cilia, which clears the mucus from the respiratory passage. Now we are going to learn non-respiratory functions of lungs. Even though the lungs are primarily for respiration, studies indicate that they have many non-respiratory functions. Some of the few important ones are mentioned below, like converting an inactive chemical precursor into its active form, like turning angiotensin 1 to angiotensin 2, which help in raising of blood pressure. It is also an essential site for degrading or inactivating important vasoactive chemical mediators like bradykinin, serotonin, and norepinephrine. The bronchial mucosa also contains a small cluster of neuroendocrine cells, also known as Golgi cells, that can secrete several factors including catecholamine and polypeptide hormones such as calcitonin, serotonin, and gastrin-releasing factors like bombicin. Then pulmonary epithelium acts as the first line of defense for the inspired air. Then tissue preparation. Proper ethical approval is obtained before the collection of lung tissue. The lung is identified, dissected en bloc, weighed, and labeled. Later, the lung is perfused with 10% formalin through the trachea to the physiological peak inspiration level. Underinflation or overinflation should be prevented. This helps in proper assessment without any artifacts and over or under the judgment of tissue structure. All the lobes of the lungs are identified, searched for any lesions. The tissue is washed well, fixed in formalin for at most almost 25 hours. Once the tissue processing is completed, it is taken to the next level of tissue embedding. The lung organ should be placed in the tissue cassette with the ventral side facing the tissue cassette. The dorsal side of the lung will be facing the open or upper side. This position guarantees the appropriate tissue section level. Post-fixation tissue trimming is a crucial component to get the best sections. If the identified lesion is too big or small, they can be isolated and embedded separately. It is essential to include the lesions of related lymph nodes for histological evaluation, which will help in turn to understand the extent of metastasis of the lung tumor. Then histochemistry and cytochemistry. Histochemistry of the lung. Histochemical analysis of the lung can be carried out using lectin histochemistry LC or immunohistochemistry or IHC. Both methods can be performed on tissue slides. The normal lung cells can be typed as type 1 and type 2 pneumocytes based on histochemical analysis using lectin typing, like glycotyping is there. The alveolar macrophages in the normal lung are positive for many N-linked saccharides, namely N-acetyl galactosamine, N-acetyl glucosamine, terminal beta D galactose, and sialyl groups. Some specific lectins can be used as a marker for cell types such as Ptolychis biflorus agglutinin for branchial epithelial cells, Triticum vulgaris for type 1 pneumocytes, and Hippisterum hybrid or Macula pomifera lectins for type 2 pneumocytes. Alveolar macrophages are anti-CD68 positive, and alveolar lining show positivity for cytokeratin. Further, IHC and LC can provide substantial insights into obstructive and restrictive pathologies as well as in lung cancer diagnosis. Adenocarcinomas can be identified by differentiation markers that include TTF1 and NAPSIN E. Both these markers are expressed in more than 85% of the cases. Then metastasis can be confirmed by IHC staining to identify primary tumor tissues of origin. Distinct expression of CK7 and CK20 profiles, in addition to the absence of markers commonly expressed in primary lung cancer, can signify metastatic cancer. With the advent of cancer treatment through tyrosine kinase inhibitor or TKI, actionable gene mutations can be screened in lung cancer patients. EGFR mutations and ALK translocation are the most effective targeted oncogenes in non-small cell carcinomas and are now considered standard treatment procedures presently. Then cytochemistry of the lung. Cell analysis of the lung tissue can be carried out using electron microscopy and have recently included flow cytometric assays. Assessment of known specific esterases alpha naphthyl acetate esterase ANAE and butyrate esterase BUT, chloroacetate esterase CHL, and acid phosphatase ACP, intracellular glycogen PAS reaction, lipids Sudan black B reaction SBB, and iron pulse reaction can be performed by a semi quantitative cytochemical method to identify diseases of the lung. ECM modeling is the basis of pathologic changes in obstructive pulmonary diseases, and collagen alteration provides insight into the disease. The second harmonic generation or SHG technique used to quantify collagen has demonstrated effect analysis of lung diseases with a biochemically distinct presentation of organization of collagen in asthma, COPD, and idiopathic pulmonary fibrosis or IPF. Alveolar macrophage modulations using chloroacetate esterase and pulse reaction for estimation of intracellular iron can confirm the presence of known small cell lung cancer. The typical increase in CML and iron in alveolar macrophages indicates non-small cell lung cancer. Similarly, galactin 3 can be used as a biomarker in human pulmonary fibrosis as the levels are observed to be elevated in this disease. Then, as mentioned before, the conducting portion is up to the terminal bronchiole. Beyond that, with the beginning of respiratory bronchiole, is the respiratory part of the respiratory system. The respiratory portion of the lung consists of respiratory bronchiole, alveolar duct, alveolar sac, and finally alveoli, where actual respiration takes place. In the conducting zone, the air is moistened, warmed, and filtered before it reaches the start of the respiratory region at the respiratory bronchioles. The respiratory zone is where gas exchange occurs and blood is oxygenated in exchange for carbon dioxide. As the respiratory tree transitions from the conducting zone at the terminal bronchioles, goblet cells diminish as club cells increase, and the cartilage present in the conducting region is absent once it reaches the respiratory bronchioles. The sinus is directly distal to the terminal bronchioles and which signals the beginning of the respiratory part. The sinus is composed of respiratory bronchioles, alveolar ducts, and alveolar sacs. It is roughly spherical, resembling a bunch of grapes. Each respiratory bronchiole gives rise to several alveolar ducts and alveolar sacs, giving it the characteristic grape bunch appearance. The alveolar sacs are the ends of the respiratory tree and the site of gaseous exchange. Alveolar epithelium is composed of type 1 pneumocytes, type 2 pneumocytes, and the occasional brush cells. Also present in the alveolar walls are the club cells and alveolar macrophages. The alveolar cells contain the pores of cone which allow communication between adjacent alveoli. This allows air to flow from one alveolus to another, which may be beneficial if there is any blockage preventing air from entering alveoli from a direct route. Type 1 pneumocytes make up roughly 90% to 95% of the alveolar epithelia. They are flat squamous epithelia that resemble plate-like structures that allow gas exchange. Their fragile membrane allows for easier gas permeability between the alveoli and the blood vessels. Despite being the primary structures of respiration, they cannot replicate and are very susceptible to toxic injury. Then type 2 pneumocytes make up much of the remaining cell type in the alveoli, accounting for nearly 5 to 10%. Despite their low number, they are vital as they secrete pulmonary surfactant. The surfactant is necessary to maintain an open airway. It lowers the surface tension and prevents the alveoli from collapsing upon themselves during exhalation. By histology, the cells have foamy cytoplasm, which results from the surfactant that is stored as lamella bodies. Type 2 pneumocytes are also mitotically active and can replace the easily damaged type 1 pneumocytes. The type 2 pneumocyte cells can be recognized by their rounded shapes that bulge into the alveolar space. Alveolar macrophages or dust cells may be free within the alveolar space or sometimes connected to the alveolar wall. If particles make it down to a sinus, the macrophages are the last defense and generators of the respiratory epithelium. The black staining seen in the lungs of smokers result from macrophages cleaning and sequestering particles that make their way inside. The lungs are covered by the serous membrane, the pleural membrane, which has two layers, the parietal and the visceral layer. The parietal pleura of the lung is lined by a mesothelial layer with underlying connective tissue and elastic fibers. An elastin stain may be used to identify the elastic layer. Then microscopy, electron microscopy. In this section, let us get into some more detailed study of a few of the essential structures of the lung, which can be appreciated better under the electron microscope, like respiratory bronchiole. This is a region of transition between conducting and respiratory portions where the exchange of gases begins. Structurally, respiratory bronchiole is very similar to terminal bronchiole, except its walls are interrupted by numerous sac-like alveoli for gaseous exchange. It is lined by ciliated cuboidal epithelium. The cilia may be absent in most more distal portions. A small number of non-ciliated Clara cells are also present. These Clara cells become dominant cells in the distal part. The epithelium is devoid of goblet cells. Underlying tissue consists of smooth muscles and elastic fibers. Distally, the respiratory bronchioles divide, become narrower, and the number of alveoli increases. Then Clara cells have three functions like produce one component of surfactant, act as stem cells, and contain enzyme systems that detoxify noxious substances. Then alveolar ducts are there. Respiratory bronchioles divide distally to form alveolar ducts. Alveolar ducts do not have walls of their own but are created by several openings of alveoli. This terminate into clusters of alveoli known as the alveolar sac, which opens into the atrium and finally into alveoli. Alveolar ducts are surrounded by small aggregations of smooth muscle cells, collagen, and elastic fibers. Sphincter-like smooth muscles like knob embedded in type 3 collagen around respiratory bronchioles and alveolar ducts regulate air movements within the alveoli. Smooth muscles totally disappear at the distal end of alveolar ducts. Rich matrix of elastic and reticular fibers provides the only support for duct and alveoli. Then the alveoli are the specialized air sac outpouching of 200 μm diameter. These are the structural and functional unit of the respiratory system. These are the primary site of exchange of oxygen and carbon dioxide. There are 300 million alveoli in the lungs, and this gives approximately 140 meter surface area for the exchange of gases. The alveoli are responsible for the spongy nature of the lung. The alveoli are lined by flattened epithelial cells called pneumocytes with a single opening. The alveolar wall or septum is made up of three tissue components, like surface epithelium is there, supporting tissue is there, and an extensive network of continuous capillaries will be there. Then centrally it has capillaries surrounded by a vibrant network of elastin, reticular and collagen fibers with a layer of squamous epithelium of two adjacent alveoli on either side. In certain places, the basement membrane of the capillary endothelium comes in direct contact with the basement membrane of the surface epithelium of alveoli with the absence of supporting tissue, thus reducing thickness 0.1 to 1.5 μm for better exchange of gases. Hence, air in alveoli is separated from the blood in the capillary by three components: surface lining and cytoplasm of alveolar cells, fused basal laminae of closely opposed alveoli, and endothelial cells and their cytoplasm. This is called the blood gas barrier, air blood barrier. Capillaries are continuous with its endothelial cells, which are extremely thin due to the clustering of nuclei and other organelles, increasing the efficiency of exchange. Occasionally, small openings, alveolar pores of cone, like 10 to 15 μm in diameter, are seen, which equalize air pressure within alveoli and allow air movement between alveoli in case of bronchial obstruction. Elastic fiber enable alveoli to expand during inspiration and contract passively during expiration. The reticular fibers serve as supportive structures that prevent over distension and damage to the delicate capillaries and alveolar septa. Two types of epithelia form a continuous lining around each alveolus. They are type 1 pneumocytes, alveolar lining cells, and type 2 pneumocytes. Type 1 pneumocytes, like alveolar lining cells. The type 1 pneumocytes are similar squamous cells that are highly attenuated with a dense, small, and flattened nucleus. This cover most of the surface area, approximately around 95 to 97% of total surface area. Golgi complex, endoplasmic reticulum, and mitochondria are grouped around the nucleus, leaving a large area of cytoplasm free of organelles, thus reducing it to a fragile blood-air diffusion barrier, 25 nm. The thin cytoplasm shows numerous pinocytotic vesicles. The surfactant lines the luminous surface. A basal lamina covers the adluminal side of these cells. The adjacent cells are connected by a tight occluding junction which prevent leakage of tissue fluid into the alveolar lumen. During fetal development, the surfactant appears in the last few weeks of gestation and coincides with the appearance of lamellar bodies in the type 2 cells. Next, type 1 pneumocytes, great alveolar or septal cells. The type 2 pneumocytes are the cuboidal cells grouped in 2 to 3 large, a central and plumb nucleus with dispersed chromatin and prominent nuclei. They occupy about 3 to 5% of the surface area of alveoli, interspread among type 1 cells with which they have occluding and desmosomal functions. Then type 2 pneumocytes are the cuboidal cells grouped in 2 to 3 large, a central and plumb nucleus with dispersed chromatin and prominent nucleoli. They occupy about 3 to 5% of the surface area of alveoli interspaced among type 1 cells with which they have occluding and desmosomal junctions. The apical surface is dome-shaped and shows numerous small microvilli associated with surfactant secretion. This type 2 pneumocytes secrete surfactant, a surface active material that reduces surface tension, thus preventing alveolar collapse during expiration. The mitotic activity of the lining cells is 1% per day and can differentiate to type 2 as well as type 1 pneumocytes in response to damage to alveoli lining epithelium. The cytoplasm has an abundant rough endoplasmic reticulum, well-developed Golgi apparatus, free ribosomes, and a moderate amount of elongated mitochondria. A typical feature of these cells is the presence of lamellar bodies. The lamellar bodies are vesicle containing phospholipid like dipalmitoyl phosphatidylcholine 1 to 2 μm. This, when discharged by exocytosis into alveoli, spreads on alveolar surface and combines with other carbohydrate and protein containing secretory products, some secreted by Clara cells, to form surfactant, which is a tubular lattice of lipoprotein known as tubular myelin, which overcomes effects of surface tension. The reduction of surface tension means the reduction of work of breathing. The surfactant is not static but continuously being turned over and removed. The removal is by pinocytotic vesicle of type 2 pneumocytes, macrophages, and type 1 pneumocytes. Then alveolar macrophage or dust cells. The alveolar macrophages are derived from blood monocytes and sometimes by mitotic division of macrophages of the lung. They contain numerous secondary lysosomes and lipid droplets. They phagocyte and remove unwanted materials such as inhaled particulate matter like carbon, dust, and bacteria. They are present free within alveolar spaces and some in interalveolar septa like spaces. 100 million macrophages daily migrate to bronchi. The pinocytosed macrophages get trapped in mucus, transported by ciliary action to the pharynx, and come out in septum. Some alveolar macrophages also go via lymphatics to fill lymph nodes. Industrial lung diseases like silicosis is because of inhalation of silica into air sacs as tiny particles that are phagocytosed by macrophages. Silicated macrophages stay for a long time and convert silica into silic acid, which stimulates the proliferation of fibroblasts and collagen, leading to fibrosis of lung and node. A particular form of silica, like asbestos, when inhaled extensively stimulates lung fibrosis, producing asbestosis and sometimes malignancy of pleura, like mesothelioma. Sometimes macrophages phagocytose extravasate RBC in alveoli, especially in conditions like pulmonary congestion and congestive heart failure. Thus, erythrocyte phagocytosed macrophages are called heart failure cells. Then pathophysiology. The pathologies associated with the lung are diverse with significant variations in disease presentation. This is primarily due to the exposure of this organ to the outside environment. Lung pathologies can be broadly classified as obstructive lung diseases or restrictive lung disease. This include bronchial disease, infectious diseases, interstitial lung diseases, neoplasm, vascular diseases, congenital abnormalities. The restriction of lung parenchymal tissue presents chronic obstructive pulmonary disease and emphysema due to chronic inflammation. Morphologically, they lead to an increase in size and number of small fenestrae in alveolar walls, fibrovascular trabeculae breakdown, and remodeling of vesicae leading to airspace enlargement. In some obstructive diseases such as bronchitis, hyperplasia of goblet cells occur, while in bronchiectasis, the bronchi are markedly dilated. The restrictive disorders, on the other hand, are marked by the fibrous deposits that restrict lung function. Then, intestinal restrictive lung diseases are characterized by inflammation or scarring of the lung tissue or filling of the air spaces with exudate and debris. Then, extra pulmonary restrictive diseases show thickening of alveolar septa and epithelium and are associated with an endothelial injury. In its acute phase, restrictive lung disease demonstrate endothelial damage. In some disorders, they also show epithelial damage with fibrosis of the exudate and expansion of the interstitium through generalized fibrosis. Chronic destructive lung diseases are marked by diffuse interstitial changes that are more prominent than the morphological changes. Many of the chronic restrictive lung diseases, particularly in later stages, are characterized by interstitial fibrosis leading to classic honeycomb-like appearance of the lung. Then some clinical significant portions associated with this topic, like infant respiratory distress. Infant respiratory distress is the leading cause of death in premature babies. Type 2 pneumocytes produces surfactant starting around 20 weeks gestation, but it is not fully secreted until nearly 30 weeks of pregnancy. Without ample surfactant, premature infants cannot overcome the collapsing surface tension in the respiratory alveoli. Physicians hope to prevent infant respiratory distress when a patient goes into premature labor by offering the patient glucocorticoids. Then glucocorticoids stimulate the production of surfactant in the fetus and may increase its production enough to help the infant overcome any potential respiratory distress. Testing for fetal lung maturity may be performed on the amniotic fluid by several fluids, including the measurement of phospholipids in amniotic fluid like phosphatidyl glycerol or PG or lecithin sphingomyelin ratio, and the lamellar body counts like LBC. Then emphysema. The most common cause of emphysema is smoking, although it can be caused by repetitive inhalation of any foreign particulate material. Emphysema or chronic obstructive pulmonary disease is characterized by poor airflow and difficulty exhaling because of narrowing bronchioles and restriction of alveolar wall. The collapse of alveoli results in a significant loss of surface area for gas exchange. Then healthcare professionals assume that the destruction of the alveoli wall is a result of excessive lysis of elastin in the interalveolar septum. The abundance of macrophages and neutrophils that migrate to the acinis due to an increase in particulate bring an equal rise of elastate and other proteases. Alpha 1 antitrypsin deficiency also causes emphysema because of an increase in elastin. However, in this disease, it is because the deficient antitrypsin usually inhibits elastin. The next disease, cystic fibrosis. Cystic fibrosis may also cause chronic obstructive pulmonary disease. It is an autosomal recessive disease caused by the mutation to the CFTR gene on chromosome 7. This gene controls the Cl gene protein involved in a variety of cells, including goblet cells in the lungs. The defective Cl channel affects the viscosity of the mucus in the lungs. Thickening is due to increased absorption of sodium and water from the lumen. The thickened mucus disrupts the mucociliary excavator filtration function of the lungs, resulting in obstruction. One of the supporting treatments for cystic fibrosis breaks the disulfide bonds found in mucus plugs, thinning out of the septum so it can be pushed out by the respiratory cilia. Then heart failure cells. In heart failure, the heart's inability to move blood efficiently results in congestion of the lungs. The increase in pressure of the blood in the pulmonary vasculature results in erythrocytes passing into the alveolar septum. The erythrocytes are promptly taken up by resident alveolar macrophages. As the macrophages engulf any red blood cells present, they are filled with hemosiderin and take on a brown granule appearance viewable under light microscopy with staining. Hemosiderin-laden macrophages are more accurately called siderophages and are not specific to a particular disease but may be present whenever blood cells enter the alveolus. Siderophages. Then tumor staging. When staging primary carcinoma of the lung, it is crucial to identify the invasion of the elastic layer of the visceral pleura. This finding will increase the T stage of tumor. Since the elastic layer is difficult to visualize with routine stains, especially elastin stain may be used to demonstrate its finding. Here we see a picture. The lungs. See at a longitudinal section of a primary lobule of lung. This is the picture showing regions. Lungs, then a primary lobule of the lung, respiratory bronchiole, alveolar duct, atria, alveolar sac, alveolus or air cell, pulmonary artery, pulmonary vein, lymphatic lymph node, contributed by some books. Then gaseous exchange in the lung. How that happens? Gaseous exchange, like blood enters, CO2 out, O2 in into the blood, and blood out, alveoli. How that is happening, which can be seen in this picture, which can be observed in this picture. Then next picture shows lung histology. This is how lung looks like. Then next topic, brief description of major respiratory organs like tracheal system. The tracheal system, the most direct and efficient respiratory system in active animals, has tubes made of a polymeric material called chitin. Insects and some other invertebrates exchange oxygen and carbon dioxide between their tissues and air by this system of air-filled tubes are called trachea. Trachea open to the outside through small holes called spiracles. Spiracles are typically paired structures, 2 in the thoracic and 8 in the abdomen. Then periodic opening and closing of spiracles prevent water loss by evaporation, a serious threat to insects that live in dry environment. Then muscular pumping motions of the abdomen, especially in large animals, may promote ventilation of the tracheal system. Then insects have a highly specialized respiratory system which consists of a network of small tubes that carries oxygen to the entire body. This meat this respiration is independent of its circulating system. The blood does not play a direct role in oxygen transport. Then most insects breathe passively through thick spiracles. Then special openings in the exoskeleton channel the air reaches every part of the body by means of a series of smaller and smaller tubes called trachea when their diameter are relatively large, and tracheoles when their diameter are very small. The tracheoles make contact with individual cells throughout the body. They are particularly filled with a fluid which can be withdrawn from the individual tracheoles when the tissue are active and high demand of oxygen. Then diffusion of gases is effective over small distances but not over large ones. This is one of the reasons insects are relatively small. Then book lungs. Book lungs form of respiratory organ found in certain air breathing attachment arrangement arthropods like scorpions and some spiders. Then book lungs get thick, narrow form of fact that the structured plates have the appearance of closed pages of a book. The series of thin plates are highly vascular, is nightly supplied with blood. This plates extend into the internal pouch formed by the external skeleton that opens in the exterior by a small slit. This provides an extensive structure for the exchange of oxygen and carbon dioxide with the surrounding air. There are 4 pairs of scorpions and up to 2 in spiders. The book lungs are found inside the ventral abdominal cavity and connect with the surrounding through an opening. These lungs are not related to the lungs of moderately swelling ceratophagus. The folds of the book lungs help in measuring the surface surface area of gaseous exchange. That diagram is located inside the filled cavity called atrium. The unfolded pages or plates of the book lung are filled with hemolymph. The folds maximize the surface exposed to air and thereby maximize the amount of gas in exchanged within the environment. In most species, no motion of the plates is needed to facilitate this kind of respiration. The trachea may possibly have evolved directly from the book lungs because the trachea in some spiders have a small number of greatly elongated chambers. Many arachnids, such as mites and some other things, have traces of book lungs and breathe through trachea or throughout their body surface only. Then spiracle is there inside insect trachea region. Then tracheole is there, respiratory system of insect, which will be represented with the figure which is shown on the left side. Along with that, blood and air space, lamella with blood chamber, stigma region, book lung of scorpion is also shown. So that's all about this topic. Later, we'll be learning about some other topics related to this. Thank you for listening.